Okay, so am I visible right now?
Yes, perfectly.
Okay, awesome. All right, so first and all, so thank you for inviting me for this opportunity for kind of like promoting our research a little bit more. So this talk will be not about the classical bioinformatics pipeline, although it is a type of informatics applied on biological data, but it's not the classical data sets that you're used to. This is more focused towards imaging. In particular, since a year or two ago, I finished my PhD, and that was mostly on electron microscopy image analysis. So starting from I think 2 years ago in 2018, I became a full-time employee at VIB, and I was partly affiliated at the bioimaging core and at the lab of Professor Ivan Sais. And so the idea was that we were developing machine learning techniques for the imaging core to solve their imaging analysis needs. And so if you look at the complete spectrum of all the data that they are able to generate along the resolution axis, then you see that we have kind of lots of modalities available to image data sets, from low resolution basic light microscopes to more high resolution light microscopes like confocal microscopy, which is kind of like the state of the art right now in light microscopes. Then we also have the possibility to image super resolution microscopy. But in this talk, I will kind of like uh, more have a focus on electron microscopy. Um, in particular, the scanning electron microscopy type. So that's on the right-hand side on this axis in here. And the reason for this is that there's lots of challenges involved from a research perspective. So the first one is the fact that we have very high resolution that can be typically up to 5 nanometers per voxel. So one pixel, one voxel would be 5 nanometers cube in size. So that means that we have lots of detail, and for biologists, every detail matters. From experience and talking with them, I know that they are usually interested in the smallest particle that they are able to resolve with these modalities. So that's already one challenge in these type of data sets. Another one is the dimensionality of the data sets that are commonly very large, because if you have a high resolution and you want to take a picture of a relatively large field of view, then you also need lots of voxels to represent that complete field of view. So a typical data set is usually in the order of several tens to 100s of gigavoxels in size. So your classical image from a smartphone or from a usual camera would be a few tens of megapixels. Here we are talking in 3D about voxels, and it is easily a factor 1,000 to 10,000 larger. So that means that we have thousands of pixels along the X, Y, and Z axes. Another aspect, as you can see in the volume on the right hand side, is also that the data is relatively complex. So it is not the contents does not really correspond to anything that you would be used to in classical images. Like if you go outside and you take a picture, the content now is really unexplored because we don't know what we could see there. It's because we have such a high resolution, we don't really know what to expect from that data. So it is incredibly complex and incredibly detailed. Another challenge that we have is that the quality of the data is usually rather poor, especially if you are interested in acquiring data very fast. So if you have limited time to acquire the data, then you have lots of noise in the data, but also blur. Like in this image on the right hand side, these edges between these objects, they should be crisp, but due to the imaging modalities, resolution limits, you encounter issues like blur and of course also noise. So we have to be able to cope with that. And then the last and maybe most challenging problem is that limited annotations are available for analysis. So say you would like to do analysis on shape descriptors for mitochondria, because in usual textbooks, mitochondria are usually ellipsoidic in shape. From experience, we haven't really assessed that. So say you want to analyze that, then you need a 3D reconstruction of the mitochondria. But the issue is that most analysis techniques to do segmentation require labels, pixel level labels, like the one that is shown in here in black and white. This is essentially a binary mask that gives you the label whether or not a pixel corresponds to mitochondrion or not. And this is time consuming to generate labels like this, especially for more complicated structures like endoplasmic reticulum or Golgi or nuclear membranes. This becomes much more complicated. So the first step, of course, is to try and improve this data quality. As I said, noise and blur are a very important issue. So that's why we first developed a technique and a plugin for ImageJ in collaboration with the Bioinformatics core and then the Bioimaging core to restore the data properly. And the main issue is that you have state-of-the-art algorithms on the one hand, and they give very promising results, but they are not really that easily applicable on EM datasets due to the large amount of parameters and also the fact that they are slow. So that's why we on one hand, we implemented those algorithms and we made sure that there was a GPU acceleration in the backend to make sure that they are really fast and that you can also interactively tune the parameters very easily in an intuitive manner so that the biologist can really optimize the parameters in order so that he sees the content changing live. So I think this would be in a video that kind of explains this. If it is playing. Yeah, so I think it is, yeah, the response time is a bit poor, but you can see this sigma parameter changing a little bit, and over time this the image on the right hand side is changing as well. So if you're interested in looking into this plugin, you can always use it through Fiji. The link is shown here, and also the paper is referenced on top of the slides. Okay, so these are some results that we had from the image quality enhancement algorithms that we have. So on the left-hand side, you see the ground truth image. That was an image that we acquired by using a very long acquisition time, an acquisition time that would be realistic in practice. It would take days to weeks or months, even for some data sets, if we would use those same acquisition circumstances. And then on the second image is a more realistic image taken like that. You could image something like that for a large volume overnight, let's say. But you see that there's more noise in that image, and also a bit more blur. And then the usual solution is a Gaussian filter. That's the image on the right, on the third image from left. And there you see that lots of detail has been lost. And so we developed new algorithms, and then the result is shown on the right, where you can see the original structures from the ground truth image are resolved much better. It's not optimal yet, but yeah, as the degraded image has lower quality, it also becomes more difficult to restore the original ground truth image. So then on to analysis. So that's one part is about image restoration, and the other part is about the analysis. And so three of the most popular structures, cell organelles that biologists are interested in are the mitochondria, endoplasmic reticulum, and nuclear membrane. So you can see them here annotated in the different colors. So mitochondria are relatively easy to find. I remember on a conference that a biologist once told me that he would be able to explain to his 7-year-old child how mitochondria how they look like, but still there are no automated algorithms to segment them. So he was a bit surprised by that. And then you have endoplasmic reticulum. Those are all these sheets that you can see between all the different organelles, so they are much more complicated and unpredictable in size and shape. And then you have the nuclear membrane, which is always the outer envelope of the nucleus of a HeLa cell in this case. So then because it is we are working with 3D data sets, we have to develop special techniques to cope with it. Here we designed a 2D unit approach. So the state of the art are like the 3D unit architectures. So you can see this architecture below. It actually consists of an encoder and a decoder model with skip connections between it. So the idea is by applying convolution operations and pooling and upsampling operations and also nonlinear operations, the combination of all those operations, they are all parameterized, and by showing it lots of examples of input-output pairs. So the input is then a 2D patch, and then the output is a pixel-level labeling. So by showing it lots of these input-output pairs, you tweak and tune all the parameters in between of the model until it is predicting exactly the output that you would like it to do. So the disadvantage of a full 3D network is that it takes way too much time to train it, to propagate it. So we developed a 2D approach in this case that is a bit more scalable. So we try to approach it from the 2D perspective, that we first train it in the Z direction, then in the X direction, and then in the Y direction, and then we aggregate all these together to get the full 3D segmentation. And I hope then next this video will play if everything is not going. So it might be, it seems kind of laggy, so maybe I'll just show an image. So this is the full volume that you are seeing here, and then what you see now is a complete reconstruction of the of all the mitochondria in there. So the idea was that we usually thought that mitochondria were ellipsoidic in shape, and as you can see here, the shape of those objects is much more complicated. They are much longer. Sometimes they branch. Sometimes you can have cycles like that. So it's in practice it's way complicated than we actually expected. So then another domain where we did some work in is domain adaptation. So here the idea is, given some labeled data sets, source data sets, can we use that data set to make predictions on another target data set where we don't have any labels available? So that's the idea of domain adaptation. And then we developed a technique based on a unit. It's a variant of it. So you see this unit architecture on top, and then we add a reconstruction decoder that reconstructs both the source and the target's data to make sure that the feature encoder is also incorporating target-specific segmentation features. And that way we were able to come up with the first segmentation algorithm for the endoplasmic reticulum. And so here, this was in collaboration with the European Molecular Biology Lab. They were able to provide us with some ER labels, and here you can see the super complicated structure of the endoplasmic reticulum. Now there are still some errors in this reconstruction, but I think we are so close to a first usable reconstruction where we can do some further analysis on. Okay, so another research domain where we have some expertise in is weakly supervised segmentation. So the more classical supervised segmentation techniques, they rely on strong supervision that you can see on the left. So that means that every pixel needs to be labeled in the image, as you can see there. So in green are the mitochondria, and then all the remaining pixels are backgrounds. And that's really intensive in terms of labeling time. So that usually takes about 20 seconds per image on average. So an alternative is to apply weak supervision. You can do all kinds of weak supervision, for example, image level labels, where you assign one label per image, and then the idea would be that this, for example, the green image would say this image contains a mitochondrion or not, so green or red. It's a binary label per image. You can also have point level supervision where you just have some pointer, something that points you to the location of the object of interest. And finally, also I think the most preferred type of labeling for biologists, and that's just the classical Photoshop tool where you just use a brush strokes to label the pixels partially, depending on the amount of classes that you have them. And that usually takes much less time to acquire those labels. It usually takes about a factor of 10 less time. So the idea is then, can we obtain a decent segmentation with these weak labels instead of with the strong labels? Could we get the same performance approximately? And this is still kind of like work in progress, but first experiments already give us some hopeful results. Here you can see the activations of a neural network that was trained on such weak labels, on weak mitochondria labels on the left-hand side and on weak ER labels on the right-hand side. And you can see that those activations clearly point towards mitochondria on the one hand and the location of ER on the other. So now, obviously, the reconstruction accuracy is much poor. It is much more. It is a bit worse, but that's logical from this perspective because we have less labeled information available. So then a final domain where we have some expertise in right now is active learning. This is also a very promising area in machine learning. So here the idea is given some oracle, some biologists that can provide us with labels, can we select the most informative samples to make sure that our classifier, when trained on these smartly selected labels, is trained optimal? So how do we start? We start with a pre-trained unit that you see on the left side. That unit, we use it for prediction of, let's say, mitochondria. So that's the patches that you can see on top here, the predicted patches. And then from all those predictions, we come up with some sample selection criterion, and we select patches that are interesting in terms of, let's say, for example, patches where we are uncertain of the predictions. So that means those patches should be relabeled and then retrained. So we select some patches to relabel, we do some dense annotation, and then retrain the units. And this cycle can go on and on until the expert is satisfied with the results, and then you can get the final segmentation out of it. And so these are then some results that we obtained. So the classical way is that you just label everything. So that's full supervision. There you get an intersectional reunion of about 86%. So higher is better. The ground truth is pretty similar to that, but that's because it was fed with lots and lots of labels. And then we have random sampling where we get an IOU of about 73%. And there you see, because we only used 120 labeled samples, that the quality is a bit worse. But then with our proposed entropy sampling approach, our proposed active learning approach, we are able to get even close to fully supervised performance. And also on the graph on the right, you see as you use more labeled samples, our entropy sampling approach, so the yellow curve, it is the steepest one, and that's what we are hoping to achieve. So for 100 samples, with our entropy approach, you obtain more than 80% IOU, and with the random sampling baseline, you only get under 75% performance. So that's a good thing to see, that just by selecting the samples to label in a smart way, you get high performance. So then let's conclude. The main thing that we are focusing on is automating analysis in 3D EM imaging, and that concerns research areas in machine learning such as image segmentation, active learning, domain adaptation, and weak supervision. And then also, we're not only looking into electron microscopy, because all these approaches, they all scale to other modalities. So We're also looking into, for example, segmentation of liver cells in light microscopy. Very recently, we started working on spatial omics datasets and also on medical datasets like rheumatoid arthritis patients in MRI scans or spinal cord segmentation in CT data. So I'll conclude with that and attention, and I'd like to address any questions if there are any. Thank you.
Thank you, Joris. I saw one question in the chat. It's from Alexander Botsky. He asks, what are the most prominent limitations for the acquisition, storage restrictions and/or time?
I think that depends on what the user wants to do. So if the user is interested in a high resolution analysis on a large scale, then you would like to have a fast acquisition because you have to image such large data sets. So then the consequence would be, because you have to image such a large volume, that the quality of your data will be a bit poorer because if you acquire data faster, then the noise level increases. That's something that we experimentally established. If you are interested in a tiny object somewhere and you can locate it in the data, then you can settle with like a high quality sample of a relatively small volume. So in that case, I think time is not really an issue. So that really depends on the application.
Thank you, Joris.
Okay, thank you, Joris. And for the sake of time, I suggest we move on to the next speaker. So, Joris, you can stop sharing your screen. Yes. And then I invite Mainak to share his screen with us.
Yes. Okay. So thank you, Christoph, for organizing the meeting, and thanks to Alex as well, the Bioinformatics Core, for bringing us all together. I hope you can see my screen. If not, just let me know.
We can see your screen. No problem.
Okay, perfect. So good morning, everyone. My name is Mainak, and I joined the Bioinformatics Core in March this year, working on multi-omics data integration projects. So the idea was that VIB headquarters wants to, enable multi-omics data integration projects at VIB and beyond in the future. So I'm working together with, at the moment, on a proof of concept project involving many different kinds of omics data. So essentially what the project deals with is metabolic error models. And so this project is led by the VIB Metabolomics Core facility in Leuven. So it's basically the team of Bart Gesk here. So they have been working for several years on patients with inborn errors of metabolism. And the idea is that they have samples from patients with amino acid and fatty acid metabolic defects, as well as patients with mitochondrial, mitochondrial defects. So the idea is to analyze different layers of omics data from each of these patient types compared to controls, and then see, of course, what is wrong, or which pathways may be dysregulated, which mutations may be having an effect, and so on and so forth. So there are, in each of these types, there are several known mutations, genetic factors, and based on those genetic factors, there are also subtypes within each of these 3 disease categories. So as I mentioned before, the multi-omics experiments have been carried out except for the lipidomics at the VIB core facilities. And just a moment. Yeah, so we have data from tracer, which is targeted metabolomics. We have data from untargeted metabolomics, proteomics, transcriptomics, and lipidomics. So as you will see, this is very much a work in progress. And so I will present preliminary data and thoughts about how we are planning to proceed with the computational data integration, where the idea is that we look at all these different kinds of data. Of course, it's easy to say, but can be quite challenging to do. Anyway, to assess all this data and then hopefully discover novel factors, genes or proteins or causal links in terms of pathways, etc. Okay, so very broadly, of course, as many of you have probably already worked with omics data, the idea is that, well, broadly, we can think of either statistical approaches or network or pathway-based analysis. This is, of course, this is the figure which I've taken from this nice MOFA paper. So it's a multi-factor analysis paper from guys at the EBI. They have been, working on these statistical models for quite some years. So when you have patients and you have healthy individuals as controls, you have the different omics modalities. So each of the modalities are features, and of course you have the different samples. So you can look at them by statistical methods, including machine learning, etc. But of course, you need to keep in mind that your statistical is, is affected by sample size, and especially in our case. So these are the numbers. The numbers in brackets are the number of patient samples we have. Amino acid, we had 6 patients, 4 for fatty acid, 6 for mitochondrial, and 5. So that was, or that is potentially, and the statistical power is potentially an issue here. So what I'm going to show today is how I've used metabolic networks to overlay the different omics layers we have, try them out with single omics layers and then combinations to see, first of all, of course, a visual exploration, and then to go more in depth into a systems level analysis. So of course, when I started the project, as we all do, it started with a literature review. I wanted to check what are the state-of-art techniques, which kind of techniques can answer or are better at answering different biological questions. And then I also started investigating different open source as well as proprietary pathway-based analysis softwares. So these are a few names. You may recognize some of them. So the one I'm going to mostly show you today is actually an unpublished, but it's out there as a preprint. So it's an app or a software called Metaboverse, and it's a play on metabolic universe. So this is the one I'm going to show you today, but there's others I've been sort of analyzing. So there is, for example, a 3D metabolic reconstruction. It's from this is the output of a large-ish consortium of about 10 labs worldwide, which look at human and other organism metabolic networks. So this is all hand curated. Metaboverse, as I will show you, is based on the EBI's Reactome database. So whatever pathways or networks are present in Reactome gets analyzed in Metaboverse. There's also other softwares such as Omics Integrator, which is from from the Frenkel lab at MIT. This is based on a publicly available protein interaction data set. So you can, if you're interested, more towards unbiased PPI data sets and you want to look at your omics data in that context, this may be a useful tool to do that. I just wanted to mention IPA, Ingenuity Pathway Analysis. So that's a nice software. It's quite a big, heavy software from QIAGEN. And the reason I want to also mention that is VIB Bioinformatics course pays for an annual license for our users. So if you're interested in pathways and omics data analysis, do have a look at this software because it is very powerful. Okay, and the other thing was that while doing all this work in the context of the proof of concept, I had to or I'm always, sensitive about keeping our approaches as generalizable as possible, because today I'm going to only show you the metabolic disease project, but there are other ongoing projects which I won't talk about today, but we need to keep those future projects in mind as well. Okay, so coming back to the proof of concept, the metabolic disease project. So the idea here is to start with the metabolic networks, which are as genome scale as possible. And the idea is that these kind of networks provide a comprehensive data integration framework for systems biology, but also for structural systems biology, because some of these networks actually also incorporate available structural information from the PDB, but also structures of metabolites. So you can start from networks and go all the way up to things like protein-protein or protein small molecule docking, drug protein interactions and stuff like that. So right now, the idea is to start from such a representation, and then we can map everything: genes, transcripts, protein complexes, metabolites, metabolic reactions. So we have the data from all of these layers. We try to explore and analyze this integrated model. And then, of course, in this project, the idea is to compare subsets. So we group the data by amino acid, fatty acid, mitochondrial errors. We group them by known mutations, or we group them by gender, etc., and then see what are the different entities that may differ between each type or subtype of disease. So that broadly is the goal of this project. So as I said, I will show you some preliminary data of how I have used this application called Metaboverse. So this is a network data integration, as well as a very, it's quite an easy and lightweight, I would say, visualization app to look at all this data that we have integrated. It is built on the Reactome database from EBI, which means that it gives you access to more than 90 organisms, pathways from more than 90 organisms. So it includes the name Metaboverse is slightly, a bit of a misnomer, because the lab that built this app, of course, is interested in metabolism, but since they are getting their pathways from the Reactome, it is not per se, restricted to metabolic pathways. So if you're interested in, let's say, any kind of other signaling pathways, as long as it's present and curated in Reactome, you still have the possibility to use this app for those kind of analysis. Now, the good thing is that it allows us to visualize and analyze single condition or multi-condition omics data. In our case, we have multi-condition, as I said, the 4 categories: amino acid, fatty acid, mitochondrial, and control. If some of you have time series data, you can also explore that with this same app. And then the idea is that you have this big hairy ball, and based on your input data, the software will highlight certain reactions or certain pathways where there might be dysregulation based on input-output abundances to each component. As I said, it's still not published, but there is a new preprint on the bioRxiv server, which indicates that probably they have already had 1 or 2 rounds of review, and I put up the link here. Okay, so the idea is that, this is a screenshot from what it looks like. You have the network visualization here. In this case, this is what the TCA cycle, the citric acid cycle, looks like on this app. Each of these shapes is a different entity. So for example, the stars are reactions, in this case, TCA cycle reactions. The circles are metabolites. The squares are protein complexes, and the diamonds are the subunits of which constitute these protein complexes. And then, of course, you also have the genes and the transcripts linked to that as well. I think it's sort of on the edge, so you can't see it here. Anyway, so the idea is that you you take your data, you do all your normalization, filtering steps, etc., and then you basically provide log2 fold changes as input to this software. So blue means downregulated, red means upregulated, and based on the so of course in this case we are looking at metabolic reactions, and from the known stoichiometric relationships, we know what are the on average what are the input output ratios that are correct, let's say, in a healthy sample. So if your input output abundances are beyond those healthy ranges, then metaboverse is going to flag certain reactions in this bold kind of bold and violet or magenta color, which tells you that these are potentially reactions which are dysregulated based on the input output abundances. In this case, I had multi-condition data, so amino acid control, fatty acid, and mitochondrial error, and certain reactions pop up as being potentially dysregulated in this case fatty acid, not in the other. So this is in terms of visualization how you can do it. And then, of course, now I'm looking at further not just basic visualization, but programmatically accessing or analyzing and curating these what they call activity motifs. So these are motifs in the sense of dysregulated abundances rather than motifs in the sense of network architecture. So based on changed abundances, this software flags certain reactions as being potentially regulatory hotspots. And currently we are annotating the details of disease-specific reaction motifs. So what we have found so far is that for it would appear that fatty acid patients have quite significant numbers of reactions that get flagged as being potentially dysregulated. So we also know we also know some of the mutations that are present in these patients. So we are also trying to link those up together with link up the mutations to see if, for example, the enzymes which catalyze these flagged reactions are also, is there any correspondence between those? Because if we can find such a correspondence, we can potentially even think of molecular targets that could be attractive in certain diseases. Okay, so this is just some. So as I said, we can look at the single omics layers. We can look at combinations of omics layers. And here I'm just showing you two fatty acid oxidation pathways. In this case, I just took the proteomics data in a completely unbiased approach. And so there are two enzymes here which so one is HADH and the other is DECR1. So these are both enzymes involved in mitochondrial fatty acid oxidation. And as you see, there are 5 or 6 reactions here indicated with these stars that HADH will catalyze, and this gets flagged in the fatty acid patients as being potentially dysregulated. And when I contacted our wet lab collaborators with this, they were really interested because this is a completely unbiased approach. We are looking at a few thousand protein abundances. And based on the knowledge that they have about their pathways of interest, they were immediately, they were already thinking of that these might be potential targets. And from our large-scale proteomics, we now see that indeed, there is a nice correspondence between what we see and what ideas they have. Okay, another thing, well, briefly, what we plan to do is to try and set up a sort of data portal for, of course, in the first instance for this project, but hopefully we can scale it up. As I said, there are other ongoing projects which I won't mention today, but to sort of have a kind of data analysis, data storage portal. For this project, we would typically store the analysis by disease type or by mutation type. And so under each of these disease type, we should have links to the affected reactions that get flagged as regulatory hotspot. And then we annotate each of these reactions, and we annotate the constituents of these reactions. And then we link up to plots which show data either as network representation or as changes in abundances and stuff like that. And then hopefully we cross-reference these entities to the appropriate resources as well. Okay, so to summarize, as I said, this is, or as you see, this is very much a project in progress. We have every time a new omics layer gets integrated, the number of potential avenues to follow up gets exponentially larger. So hopefully we can soon sort of come to a consensus. And anyway, so I showed you a kind of network-based data integration approach. Hopefully in the near future, we'll also be, or I will also try to apply more statistical approaches to this data. One of the things, as I mentioned, is to try and set up this multi-omics data integration portal for this project, but in the future for other projects as well. And of course, as you can imagine, many programs and scripts are being written, and so hopefully we can start organizing them into smaller toolkits and put it on GitHub or whatever for use by the community. The other thing I wanted to mention before finishing is that, so we at the Bioinformatics core, as well as at VIB headquarters, are sort of definitely interested in getting involved in and facilitating many different data integration projects. So please feel free to reach out to me or to Alex, and I would be very, very happy to learn about your projects and hopefully share our experience as well to help in your projects. So I'll just end by acknowledging the people involved in this project. As I said, it was started out as an initiative from headquarters, mainly by Tony. And then on the experimental side, it's mainly the team of Bart Geskier, and my main contact there is Matthew Bird, who was a postdoc in the lab, and he did most of the sample preparation, etc. And then, of course, some of the other VIB cores, metabolomics core, Ghent, proteomics core, nucleomics core. So everything is VIB core facility driven in this project, except for the lipidomics work, which was done at the Swinen lab at KU Leuven. So with this, I'll finish up. Thank you for your attention. And as I said, of course, please ask me questions now or at any point in the future. Just send an email to me or to Alex to discuss about your data integration projects. Okay, Christoph, I'm done. And
thank you, Mainak, and thank you for highlighting the core facilities, which is very, very nice. Okay. For any questions, general, we have an email address, bits@vib.be. So that's maybe easy to remember, than a personal email address. Either way, it will be brought to the correct person. So either direct or either to bits@vib.be. Okay, so thank you, Mainak.
Okay, thanks, Christoph. So I can.
Yes, okay, great. And then, yes, and the last speaker for this session is Bert Droesbeke from Elixir Belgium. He recorded his meeting, so I will share my desktop first and play the video. Okay, I hope everyone can see my screen now. If not, let me know. And if you're not able to hear it, also let me know. It should include sound as well.
Hello, everyone. My name is Bert Droesbeke, and today I'm going to talk about Galaxy containers. I'm part of Elixir Belgium, and for people that never heard of Elixir, Elixir is an intergovernmental organization that brings together life science resources from across Europe. The Belgium node, which is located here in Ghent, builds on existing national resources and people and covers all aspects of data. starting from data management planning all the way to reusing, as seen here in this data lifecycle diagram on the right. My title is Galaxy Containers, so it's obvious that I will first say a word about Galaxy itself. Galaxy is an open source web-based platform for data-intensive biomedical research. It's suitable for researchers without informatics or system admin expertise to actually perform very complex computational analysis and to visualize their results. Galaxy is using shareable workflows and captures information during the workflow run, making reproducing results very easy. With thousands of tools available nowadays, Galaxy is present in many science fields. We like to see Galaxy as a one-stop shop, starting from processing raw data and ending with the generation of graphs and images for publication. Elixir Belgium has set up its own instance, usegalaxy.be, which is running on the cloud infrastructure of the VSC, the Flemish supercomputer. This is a screenshot of the famous interface of Galaxy, with here the tools panel on the left. The center console for tool, data, and workflow interactions, which you don't see here right now. And then on the right, all the input and output files that are available in your history panel. The Galaxy Training Network, or in short GTN, focuses on providing training materials for Galaxy. And this not only to train researchers, but also admins and trainers themselves, which is also important. GTN also aims to make it easy to find other Galaxy trainers and to share and discover training resources available for Galaxy. All these training resources are spread over 111 tutorials and divided in 16 main topics. And these are all available on GitHub. Which also makes it easy to actually contribute, since this is a community effort. If you visit the website, you immediately see that based on the topics here, that the tutorials cover many fields in science. If you click on one of the topics, like for example assembly, you will find that the content for each tutorial is here available on the right, with slides, hands-ons, but also the input data sets. And those input data sets are all put on Zenodo, which is nice because that makes them citable and also brings version control. And this data that is used in the tutorials can be harvested from Zenodo automatically into Galaxy. And they actually put it up a system to make sure that the Galaxy Training Network data libraries on, for example, usegalaxy.eu are always in sync with the trainings. And next to data for the tutorials, you also have the discussed workflow, possible tour, and then most importantly, where you can actually run the workflow and follow on which public instance. So that's here visualized. But it is not always a guarantee that a public instance will actually contain all the tools, and especially the correct tool version for the corresponding workflow that you want to run. And that brings me actually to the next slide. So there are some limitations of using public instances, because compared to the data libraries that are automatically up to date with for example, the training network, for tools, it might not be, and it's most of the time the administrator that has to update those, and it's like a manual action. And yeah, a second point is that public instance might be down for maintenance, or because there is some problem, or if you want to use the workflows in the tutorials to actually run your own data, it might be cumbersome to transfer all your data to a public instance, especially if we're talking about big files. So for this, a fail-safe or second option, whatever you want to call it, is made available: containers. And containers is something that everyone by now probably already heard of, and is gaining popularity by the day. You can see a container basically as a box that does not only contain your tool, but also the libraries, dependencies to run the tool itself, making it perfect for preservation and, yeah, in other terms, reproducibility. So the container itself is built from an image which can be seen as a blueprint. The most popular of these container systems is Docker, and it runs basically on most user operating systems, including Windows, Linux, and Mac OS. And yeah, this means that once you have Docker installed, you can run every container that is available. And we were lucky that the lab of Björn Grönig built such a container with a bare-bone Galaxy instance inside that can be run by one single command that is seen here in this slide. And yeah, if you run this command and you visit your browser and go to localhost 8080, you will have access to your own local Galaxy instance. Because, yeah, with this container approach, we want to tackle the tool version problem. We actually went for a workflow-centric approach. So that means that we extract the tools and dependencies that are needed straight out of the workflow itself, instead of using a separate tool list file. So this way, the container will always be able, in most cases, to run the workflow. This is done using Ephemeris, which is a Python library to manage Galaxy. And as seen here, yeah, everything what we need is available. This is an overview of the basic principle. So we start off with the Galaxy base image, on which the tools are installed using the workflow file. But next to that, we can also bring in the data using data libraries in Galaxy. But this is not always preferable, because depending on the use case, the data can be quite big, and that will also make the image of the container quite big. So yeah, this can also be preferably done on container run to import actually the sample data. The concepts and their corresponding build scripts described before were generalized and documented in the GitHub repo Biocontainers for Training. The next step was to standardize the Galaxy training materials. So they would work with these build scripts. This allowed us to have a working Galaxy container for every topic. So if you now visit a topic on the Galaxy Training Network, there will be instructions available in how to run your own instance. So then it will look like this with the workflows available and all the other stuff for the tutorial pre-installed. So a custom Galaxy instance specifically made to run those specific tutorials. There are some pros and cons about running your own Galaxy container to do trainings or analysis. And yeah, the advantages are clear. If you run your own instance, of course, you always have full control and admin access by default. And you're also in charge whether it is running or not. So this can even be offline, something that might come in handy some places in the world. I will touch on that later. And since your data is kept locally, you don't need to worry about privacy. So containers can also be built automatically from a GitHub repo, which makes it always up to date if needed. And last but not least, you want to import lots of data, then this will go quicker in a container that is locally than if you want to upload it to a public instance. Of course, there are also disadvantages, and one of the big ones is that there is a significant performance drawback. And in certain use cases, also the image file can be quite big. So the base is 3.4 gig, and everything what is being installed on top might bring it to up to 30 gig or more. And negative point is that some tools just need additional care to actually be working on Galaxy. And since the tools are automatically installed, some steps that are needed are not done. So it might be that the tool is not working. And since this way of doing things was documented carefully, this possible to apply this easily on other use cases besides the training network. And so I'm now going to talk about some examples where this concept came in handy. In addition to the automatic building of the Galaxy container, we also added the possibility to actually automatically execute workflows on top of that Galaxy workflow. And this was done using BioBlend. It's a Python API to connect with Galaxy to control it. And by basically mounting the input data in the container and mapping it to the input steps of the workflow, it is possible to start the workflow and get the outputs, and this all automatically, basically starting from only a workflow file. Another example is the SARS-CoV-2 analysis workflows. So yeah, in the beginning of this year, when COVID-19 started affecting our lives, the Galaxy team immediately combined their forces to make SARS-CoV-2 optimized workflows. These workflows are suited for genomics, cheminformatics, and many more analysis. But yeah, as said before, if you, for example, the raw reads for COVID-19 analysis might still contain a human reads, which is not preferable to be uploaded on a public instance. So this is where these containers come in handy, because you don't have to worry about that, like mentioned previously. this was also set up very quickly since basically all the knowledge was already there. Another example is eBioKit Africa. Reliable internet is often taken for granted, but yeah, there are many countries in the world, and yeah, Africa is definitely one of them, where yeah, infrastructure is just a big problem. And with containers, it is basically possible to download the containers in advance onto your system and to then run them locally, yeah, without the need of internet. The last use case that is actually still going on is the ENA upload container. Within Elixir Belgium, we made at this moment 2 tools to improve the uploading of COVID-19 read data to ENA. And the first tool removes in a very easy and convenient way the human reads from the raw reads. And also another tool is then able to submit these reads together with its metadata to ENA. That last functionality is based on ENA upload CLI, something we worked together on with the lab of Björn Grönig, and this actually makes the whole process of submitting data to ENA a lot easier, because people that are familiar with this probably know that it's quite cumbersome to XML files, and you can also not use ask a researcher to upload its data to an FTP server. So this is all covered in the tool and made available through an easy user interface. And since this is all packaged in a container, this can easily be deployed and used in other countries, so they don't have to worry about privacy. How to do all this is documented on the new website that has recently been put online. It's rdm.elixirbelgium.org, and it gives general information about data management and also a step-by-step guide for the COVID-19 submission using our Galaxy tools. And soon there will be a screencast, so probably you will hear more from this in the future. Thank you for listening, and if you have a question, don't hesitate to ask. I have something to add there. The COVID-19 raw reads that were from Belgium uploaded were done using this container and this approach. So it is working. So like I already said, if there are questions, I saw that there was actually a question in the chat from James Collier. I'm not taking over your job, Christophe, but I will read it. Do you have integration with other container type environments like GUIX or Nix? I do not have experience with those container integrations, and also don't immediately see the purpose, because one of the big goals of using the containers was to actually bring it easy to users, that he could easily set up their own Galaxy instance, no matter whether it was now Windows, Linux, and to make it as simple as possible. And I don't know whether these approaches are for the average user.
Thank you, Bert, for the presentation and for the question. If there are any other questions, by all means, pop a message in the chat. James has another comment.
Yeah, so where tools are not automatically configured correctly. I can actually give here a very specific example. For example, the Galaxy tool that we made makes use of a brokering account to actually upload to ENA itself, and the instance has to be set up in a specific way that actually, for example, the account credentials are available as, yeah, in this case now as environmental variable, and yeah, this is something that needs to be taken in mind when the tool is installed. But yeah, I can't give specific other examples, but I know that, yeah, sometimes with tools, specific links with databases need to be made. And for sure, if you know that in advance, those things can be taken care of when the container is being built. But those are then like, yeah, manual interventions. Okay. Yes, we are working on that. So they could indeed be supplied at runtime. And I think the upcoming version of our tool will work on that. But yeah, I can give specific names of tools that didn't work out of the box, but within the Galaxy training network, I definitely came across some problems with tools that, yeah, they said to me like they need some extra care, but sadly I don't know the tool names itself.
Okay, thank you, Bert. We are a bit over time, but really doesn't matter. Thank you to Joris, Mainak, and Bert for talking in this first bioinformatics seminar sessions. Now, everyone interested in bioinformatics and especially software development, there is also a Research Software Developers Day. You will find the link in the chat. This is an event organized by us, by Belgian research software engineer, and you'll find more information on the link I posted there. You'll find not the program, but you can find all the information there. And like James put it in the chat, yes, more information on the website of the Belgian Research Software Engineers as well. Thank you, James. Now, with this, I would like to close this first bioinformatics seminar session. Now, if people feel inspired or they have a certain urge to present, well, by all means, let me know. Then we will try to plan the next session. It will be online as well. I think we will hold online sessions for quite a while. Maybe somewhere in 2021, we can switch to live sessions, but that's for the far future. So if you have any questions, you want to present something, email to bits@vib.be. That's the general email address of the Bioinformatics Core. And with this, I want to thank everyone, especially the speakers, for joining. And with this, enjoy the sunshine, because the sun is shining today. So enjoy the rest of the day, and hope to see you for the next event. And I'll keep you posted with an email, and hope to see you there.